So welcome everyone. This is the new EUFORGEN webinar series, and it's the first webinar. It's on estimating effective population size for forest genetic resources conservation. The aim of this webinar is to bring together all the experts on this topic to present their views and to see if we have the effective population size as an indicator for an effective forest genetic resources conservation. This webinar is organized by the European Forest Genetic Resources Program. I'm Anna Maria Farsakoglu. I'm a researcher at the EUFORGEN Secretariat, and today I will be your moderator. The outline of the webinar will be 3 presentations, 15 minutes each presentation, and 5 minutes of questions. Just a clarification here that the 5 minutes questions are only if you require some clarification based on the presentation, because the main discussion will be after the end of all the presentations. Then we will open the floor to everyone and we will have an open discussion. So today we have with us Juan Jose Robledo-Arnuncio from INIA in Spain, Son Hoban from the Morton Arboretum in the United States, and Bruno Fady from INRAE in France. And I would like them to introduce themselves one by one and say a little bit about themselves. So I will start with Juan Jose, please.
Hi, thank you, Ana Maria, and hello to everyone. So I'm Juan Jose Robledo-Arnuncio, and I am a research scientist at the Spain's Institute of Forest Science in Madrid. And I'm a population geneticist working on demographic and evolutionary processes that affect contemporary adaptation of tree populations, with special focus on the interactions between gene flow and local adaptation.
Thank you, Juanjo. Then Son.
Hi everyone, my name is Sean Hoban. I'm a tree conservation biologist at the Morton Arboretum, which is a botanic garden outside Chicago. I am trained as a population geneticist in trees. My research program has moved more into applied conservation and conservation for policy and management over the years. And so my lab's focus right now is basically providing the knowledge needed for conservation action and for policy. We also have an interest in demographic change over time, communicating to policymakers, hybridization, as well as providing advice to botanic gardens about ex situ gene conservation.
Thank you very much, Son. And last but not least, Bruno.
Hello, everyone. Bruno Fady. I work out of the National Research Institute for Agriculture, Food and the Environment in France, INRAE. My lab is in Avignon. It's a lab called Ecology of Mediterranean Forest, where we study the dynamics of forests from different perspectives: genetics, ecophysiology, fire science, and entomology. My training is in ecology and population genetics, and I'm interested mostly these days in how gene diversity should be taken into consideration in conservation. Thank you.
Thank you very much, Bruno. So I will stop sharing my screen and we can start with our presentations. So Juanjo will be the first one to present us the effective population size in forest genetic resources conservation with some theoretical and practical considerations. Juanjo, the floor is yours. And you have 15 minutes.
Thank you. I will start the timer. Can you see the presentation? So I assume you can all see the presentation now? Okay.
Yes.
So first of all, thank you again, Ana Maria and Michele and EUFORGEN for organizing this seminar and for inviting me. And thank you all for coming. I'm sure we are going to have a lot of interesting discussion. And I see many people here that probably have much to say. So I encourage all of you to participate in the discussion. So What I'm going to do first is to make some general theoretical considerations on what is effective population size and why it can be useful in conservation genetics. And then I will discuss practical questions related with the estimation of effective population size in realistic demographic settings that are typical for many of the species of trees that we work with. So my presentation is based largely on a paper that we published last year, co-authored with Luis Santos del Blanco and other colleagues at INIA, at INRAE, and at Nottingham University. So what is NE? NE, probably all of you have heard this definition, is the size of an idealized right Fisher population that shows the same value of some specified genetic property as the real population. So imagine we have a real population of census size n, and we can, for instance, describe or observe a genetic process like drift, which is interesting in conservation genetics because it determines the loss of diversity. We can observe, for instance, the allele frequency variation across generations. So then NE would be the size of an ideal population that shows this same drift pattern as the real population. And by ideal population, we mean a population that is isolated, that has random mating, constant size, Poisson distributed offspring number, non-overlapping generations, and equal sex ratio. So in any departures from these assumptions, from these ideal characteristics will result in a difference between the census size n and effective population size n. In general, n will be lower than NE, but not necessarily in all cases. So then why considering NE in forest genetic resource conservation? Well, FGR aims at preserving genetic diversity and evolutionary potential, mostly avoiding inbreeding and genetic erosion. And it's NE rather than the census size n that determines these evolutionary processes. And typically, we know that in many cases, NE is lower than the census size. From many empirical studies, we know that the median estimates of the ratio is about 0.1 or 0.2, but highly variable among populations and species. So the lower NE is, the higher the inbreeding rate and higher drift, and eventually the higher inbreeding depression, genetic diversity loss, and the less efficient natural selection. So you probably have also heard about the 5500 rule. Probably Sean will talk a bit more about this. But in conservation genetics, we usually consider that if the effective population size is larger than 50, we would minimize the short-term inbreeding, below 1% per generation. And if the effective size is larger than 500, genetic variants available for selection wouldn't be lost because it would be replenished by mutation at the same rate that is lost by drift. So all this is theory. And the question is, for sure, if we can estimate NE accurately, it will be mostly very, very interesting in conservation genetics because it will pinpoint as populations at risk of inbreeding and diversity loss. But the question is whether we can estimate it accurately in practice. And the first questions that we face in practice is that we have to choose a target population. And if we have an isolated populations, for instance, a genetic conservation unit, then it's clear that the target population will be the same as the real biological population. But in other cases, in many other cases, we will have a set of populations that are potentially connected by pollen or seed dispersal. And in this case, maybe we have an administrative limits that define our target populations. Again, it might be a conservation stand, a genetic conservation unit, etc. But we would have to think whether this is actually the most meaningful target population to define from an evolutionary perspective, or rather if we should be looking at the whole meta-population. And also in trees, as you know, we have many instances of very widespread populations, more or less continuously distributed of over hundreds or even thousands of hectares. So in this case, we might have a genetic conservation unit that maybe does not match the entire distribution of the population, but is rather limited within this larger population. In this case, we would have to ask whether it makes sense to set the target goal for any estimation in this smart part, or whether we should be looking at the entire stand. So once we have a target population to estimate any, we have to think about a population genetic property that we would like to analyze. And if we are talking about inbreeding, we will have the inbreeding effective size. If we are talking about allele frequency variance, we can define the effective variance size. We have the additive variance effective size, the linkage disequilibrium effective size, or the coalescence effective size, and others. An important question here is that if we have an isolated population, a discrete isolated population of constant size, all these definitions converge, but not generally otherwise, okay? So these values can be different if we have, for instance, a metapopulation. So we have to think in general in conservation, as I said before, we will be probably interested in the inbreeding effective size and the additive variance effective size, which maintains the evolutionary potential. So what is our main interest is one question, but on the other hand, what can be estimated with the means at hand? Not necessarily the inbreeding and the additive variance effective size are easily estimatable, and usually they are not. So then we have to choose an estimation method, and any can be estimated using pedigrees or relevant demographic information and reproductive information. And I will say a bit more about this later. Or in general, and most frequently, people are using genetic methods to estimate effective population size. And it's important to notice that both the estimation method that we choose and the sampling design will determine the temporal and spatial scale of any, and this is independent of estimation error, okay? So I will talk about genetic estimators because, as I said, especially in trees, they are in natural tree populations. They are very frequently used for logistic conveniency. And we have two large groups of estimators. One that are historical, that calculate the geometric mean effective size per generation over many generations and typically incorporate the mutation rate. And we have contemporary estimators that estimate the effective size of the current or the few previous generations. And these contemporary estimators are generally the ones that are interesting for monitoring in conservation genetics. Although, of course, historical ones can also be interested. If, for instance, we want to know if a small population has been small for long before human intervention or not. I will focus on contemporary estimates as potential useful indicators or verifiers in conservation genetics and monitoring. And we can distinguish two other big groups. One are the temporal methods that are based on temporal allele frequency changes in time and require at least two samples. I mean, they require sampling in at least two different time points, which may be rather difficult in trees. And then we have the one sample methods, which require samples taken in a single point in time, and which are the ones that are most frequently used in conservation genetics using genetic markers. One sample methods have a lot of variants. We have the linkage disequilibrium method, the heterozygote excess method, CSIC frequency method, summary statistics method, and others. And you probably have heard about many of these. I will focus on the first one, the linkage disequilibrium method, because it's the most widely used and because in many cases behaves statistically similarly to other methods, even better in some cases, not always, but of course, each one of them has its own set of assumptions and potential issues in practice. So now I will discuss some practical issues specifically related with demography of trees. As you know, trees have frequently very long distance gene flow, which means that the genotypic distribution over a metapopulation is not only affected by drift, but also by gene exchange among populations that are potentially genetically divergent. So what happens in this case? One potential way to look at it, it's as Ryman and colleagues did, is to look at the realized effective size and in a simulated metapopulations. In their case, they consider 10 subpopulations with a local size of 50 and a migration rate of 0.2. And what I find really interesting in this study is that what they found is that once you have equilibrium in the metapopulation, drift migration equilibrium, what you see is that the inbreeding local effective population size and the additive variance local effective population size converge to the metapopulation inbreeding and average and additive variance effective population size. These are the top lines here. By contrast, what you see is that the linkage disequilibrium effective size and the variance effective size are much lower and remain close to the local census population size. What this means is that local inbreeding and local additive variance, the temporal additive variance, are determined not by local demographic process, but rather by the entire demography over the metapopulation, okay? And on the other hand, linkage disequilibrium is more determined by local processes, okay? The higher the gene flow rate, of course, the higher the local effective population size will be. And if you have a migration rate of 1, of course, any kind of local effective population size definition will converge with the global one. So here what is interesting is that the usual target in conservation genetics is the inbreeding and average additive variance effective population size. But what we usually estimate out of statistical convenience is this, the linkage disequilibrium and the variance effective size. Other issues that trees are usually widespread and establishing very continuous stands where there is isolation by distance. And we know since a long time ago that in this kind of populations we have a different genetic process, which is isolation by distance, and that can be characterized by the neighborhood size, that is the inverse of the probability of identity by descent of 2 uniting gametes. And this parameter determines the spatial genetic structure within the population, but it has an unclear relationship with the loss of genetic rate at the population as a whole. And the issue is that when we estimate effective size using, for instance, the linkage disequilibrium method, what we are actually estimating is the neighborhood size, if the sampling scale is small relative to dispersal scale, or a quantity between NS and effective size otherwise. Then we have the issue that trees are heteroparous and they grow in age structure populations. And actually, we have no clear, there is no well-established expectation of one sample contemporary estimators of NE with age structure. The expectation depends on the distribution of samples across age classes, on age at maturity, and on longevity and others. Then if we sample a single cohort of offspring, actually what we get is something that depends not only on the effective population size, but also on the effective number of parents contributing to the cohort. On the other hand, if we sample only random adult samples, the presence of age structure will result in an underestimation of NE. Then, except for endangered species or marginal populations, trees typically have enormous population sizes, and the problem is that the signal to estimate NE is inversely proportional to population size. So to get reliable estimates of NE with the linkage disequilibrium method, we require samples of the same order of magnitude as the effective size. And when N is large and the sample size is small, we typically get a bimodal distribution of estimates with many infinite ones and many that are orders of magnitude smaller than actual values, okay? On the top figure, you see a case with a small population where estimates work better. Here are some examples that we did of estimating any contemporary NE in large populations in Spain, 19 large continuously distributed populations with small samples of 10 to 28 individuals, one large population with the sample size somewhat larger, 150, and then a small isolated stand where we sample 100 out of the 300 standing individuals. And what we saw, it's in the 19 large population, we got a median estimate of 300, which corresponds to a ratio between NE and N, very, very small, 10 to the minus 3 to 10 to the minus 2. And interestingly, this effective population size estimate corresponds to the neighborhood size that we estimated independently. In the other large population, here you see that the effective population size estimate is also around 300 for different subsample sizes. It was, in all cases, around 300 individuals. And it also was somewhat larger, but in the order of magnitude of the neighborhood size. Finally, in the small population, we got an estimate of 29 for NE, which corresponds to a ratio between NE and N of 10 to the minus 1, which is a bit more normal. We believe that all other estimates in the large populations probably are strong underestimates because of the small sample sizes and because of spatially restricted sampling in the large populations. So to conclude, some conclusions or things that a responsible scientist should think about before giving advice to conservation managers, and is that except for small isolated stands, precise definition and accurate estimation of contemporary NE is very complicated in trees. Be extra cautious and disclose known limitations when working in a risk assessment or decision-making context. Consider carefully the demography of the target system before proceeding with the method choice. Check its assumptions and before sampling and estimating. Remember that under gene flow, the rates of local inbreeding and additive variance loss may be influenced by regional genetic processes. But on the other hand, marker-based NE estimators, such as the LD or temporal estimators, largely reflect local processes. Small and spatially restricted samples may bias downwards marker-based contemporary NE estimates, even without gene flow. And always report uncertainty estimates for both N and NE. Okay, and from the point of view of conservation managers, be clear about the conservation genetic goals, define the target spatial scale, noting that the scales of management and relevant population genetic processes may differ. Consider that except for isolated stands, genetic process determining tree inbreeding and evolutionary potential may be operating beyond population boundaries, artificial population boundaries. Unbiased and precise estimation of genetic parameters in widespread tree populations is very difficult statistically. Market-based contemporary NE estimators tend to reflect local processes and suffer substantial bias and variance, so they may be riskier than a carefully chosen value of the ratio between NE and N, and also a carefully chosen value of the census size as indicators of the risk of inbreeding and evolutionary potential. Of course, do not neglect demographic indicators with special attention to recruitment, and consider monitoring inbreeding, mating system, gene flow, and genetic variation directly. So sorry for the delay, and with this I conclude with thank you to all of you and to my co-authors and the funding agencies of this work. Thank you.
Thank you very much, Juanjo. So for those that were not with us since the beginning, we will allow 5 minutes of clarifications after each presentation, and the final open discussion will be at the end. So if there are any clarifications regarding Juanjo's presentation, you can raise your hand now or type your question in the chat. I think everything, soon, please.
Just to break the ice, you talked about migration between populations and metapopulations. Can you clarify what level of migration between populations will correlate to turning a population basically into the any of the meta population. So what kind of migration are we talking about?
Okay. Yep. So that's an interesting question, and one have to distinguish two things. One is actually the realized effective population size. This is what is actually happening in the system, independently of the estimation. And from that perspective, what we see is that the higher the migration rate, actually the local effective population size will progressively converge to the metapopulation effective size. So, and this is more or less continuous a process. Well, we could maybe later with more detail, we can look at some of the graphs of the people that have studied this. But the important thing is that it will slowly converge to the metapopulation effective size. But from the point of view of estimation, what we see is that if migration rate is below about 10 to 20%, with the linkage disequilibrium method, you get an unbiased estimate of the local effective population size, okay, the local one. So the bias is relatively small if what you are estimating is the local effective population size. About 10 to 20% of the migration rate, then the bias in the estimation of local effective population size increases, and actually what happens is that you tend to obtain an estimate closer to the global metapopulation size without never reaching it.
Thank you.
Thank you. So now we are going to the list of the attendees. I would like to ask you to briefly present yourself when you will take the floor. So I see Robin, I suspect it's Robin Waples. The floor is yours.
Okay. Thank you. That was very nice presentation. My comments just had to do, I think, related to Sean's recent comment. And it's really important to think at the start why you want to estimate effective size. So you talked about bias, say the single sample estimators. But that depends on what quantity you're interested in. If you're interested in a global or metapopulation or species-wide effective size, then even a very little migration won't affect the local LD estimate much. It will still estimate the local demographic genetic processes going on. But even a very small amount of migration is enough to keep alleles moving among subpopulations. So the total genetic diversity in a local subpopulation is very strongly influenced by even small amounts of migration. So if you're interested in local processes, what are going on within your local population, then the single sample estimators may actually be quite good because they aren't very sensitive to the metapopulation processes unless migration rate is really pretty high. But if you're really interested in genetic diversity, how many alleles, heterozygosity, all those sorts of things, then you really have to be careful about even very low levels of migration. So if that's what you're interested in, then I would agree that the single sample estimators that are estimating local NE, that would be a bias compared to what you're interested in that case. And I think that's one of the major points that Niels Ryman and his colleagues were making in the 2019 paper that you cited. So again, I would just emphasize, which I think you stated at the start, it's really important to think about what you want to do with this information about effective size, because that will determine the sorts of analyses that would be useful for your purposes.
Okay, yeah, so thank you, Robin, absolutely. And I'm really happy that you are here because I can't really, probably won't be able to say any more than that you can yourself, that you have looked at these questions much more carefully and deeply than any of us. So yeah, for sure. And especially just in addition, if what we are interested is in, for instance, evolutionary potential, I think that it is even more important to be looking at the global scale, even determined by not non-local processes. But thank you for the clarification.
Thank you very much. We will take for the moment one more question because we are running a bit out of schedule. So Manasvi Bandari, just a brief question, please, and then we will move on with the next presentation. We will have more time at the end after all the presentations. We will have a lot of time.
Hello.
Hello.
I am audible. Yeah, this is my daughter. The talk. I listened Dr. John Joe presentation and very nicely presented his work on the finance minister. I would like to ask that if the finance minister is most obvious is gregariously distributed species, and in some of the temperate forest in the Himalayan region of India are mostly the species are in the mixed forestry species are in the scattered form. So, what is your opinion on the means how to carry out the population sampling so that we can represent the number of individuals from the scattered tree species in so that it can give reliable information on the population genetic estimates. Thank you.
So, well, I couldn't hear very well the question. I think the question was what's the kind of distribution Pinus pinaster has and how would you sample in a scattered tree species, or is that okay? Well, Pinus pinaster is scattered, but I mean, on the other hand, it has very large populations, okay? So thousands of hectares each. And how to sample there? Well, it's really hard. I mean, when you have a large population, any kind of statistical exercise to estimate any will be really difficult because the drift signal will be tiny. So, and it will be smaller actually than the sampling noise. So you will need huge sample sizes to get accurate estimates based on genetic methods. In that case, it might make more sense to use reasonable and biologically meaningful estimates of the census size coupled with, for instance, fecundity variance estimates and gene flow or gene immigration estimates. That's my opinion. We probably can discuss more about it later in the general discussion because it's a very important topic for trees.
Thank you very much. Yes, indeed. And I think we are all waiting for this open discussion. So let's move on with our presentations. The next one is Son, and he will talk about conserving genetic diversity of species without DNA data, imperfect but useful indicators. Please, the floor is yours.
Thank you, Ana Maria. Thank you, everyone. You can see my screen. It's okay. Let me know if you can't see the screen, but I'll just begin.
Yes, we can see the screen. Perfect.
Okay, great. Thanks a lot. So I'll be talking about these indicators for conserving and monitoring genetic diversity, especially without DNA data. I acknowledge they have some imperfections, but I hope to show today that they are useful nonetheless. And I'm presenting on behalf of many collaborators that I've worked with over the past 4 years on this topic. What is an indicator? An indicator is a simple summary of the state or trend of a complex system. They're especially used in ecosystems, in ecology, in economics, etc. Indicators should be used for several things. They should be used for prioritizing what species or populations need intervention, for setting quantitative goals that we want to aim for the conservation outcome. They should ideally help guide management actions to reach those goals. They should help document and report change over time. Are we making progress toward those goals? And they should be used for communicating to the public and policymakers about the importance of this system, about the importance of biology, and again, about whether we are making progress towards our goals. I've mostly worked on indicators in the context of international policy, especially the Convention on Biological Diversity, but also others, including the EU Habitats Directive and the FAO. We also want indicators to be useful not just to policymakers, not just for documenting and documenting a biodiversity decline, but also for reversing that decline. So indicators should be helpful for management decisions for where we need to do restoration, for example, or connectivity most urgently. And they can also be used for endangered species legislation, such as allocating funding among species. Indicators should have certain qualities, such as sensitivity, detecting changes. They should be, of course, be biologically relevant. They should be simple and able to be explained to non-scientists to some degree. And they should be feasible in all countries, that is affordable, and they should be legitimate, that is accepted by stakeholders. If we wanted to come up with genetic diversity indicators from scratch, what kinds of things might we focus on? My colleagues and I thought about this, and there are other answers to this question, but the answers we came up with were, we want to prevent the loss, we want to maintain sufficiently large populations for all of the reasons that Juan Jose mentioned, drift, inbreeding, and adaptation. We want to prevent the loss of distinct populations and their local adaptations to help the overall species adapt, and when possible, monitor using DNA, monitor things such as hybridization, gene flow, etc. This would address the two main components of genetic diversity within and among populations. So this led my colleagues and I to propose 3 indicators to the Convention on Biological Diversity relating to populations above an NE of 500, relating to populations maintained relative to a historic baseline, and relating to the amount of genetic monitoring or genetic knowledge that's being developed and used for management. Focus on effective size today for this talk because it's the topic of our webinar, but I want to mention the other indicators are also very important, especially maintaining distinct populations. So as Juan Jose mentioned, NE 500, there's a theoretical and practical basis to this. We know that once populations are much below 500, as shown in this graphic, genetic diversity decline due to drift increases very rapidly, which then can lead to inbreeding in the extinction vortex. Natural selection is less efficient, etc. So it's important from relevancy, but I think it's also a concept that some stakeholders can understand because it's already in use in sectors such as forestry, agriculture, fisheries, and conservation, such as the designation of genetic conservation units. NE and the NE 500 goal has also been mentioned under the EU Habitats Directive, including as a possible waypoint for the favorable reference value or favorable conservation status. Effective size, and the effective size 500 is also more and more frequently mentioned under the US Endangered Species Act as a measure of whether current populations are resilient or not. And even for trees, such as this widespread dominant tree species, whitebark pine, recently listed under the US Endangered Species Act, NE 500 is used as a goal for restoring populations and also identifying populations that are suitable with sufficient genetic diversity to sample seed from. As Juan Jose mentioned, we are interested in the contemporary NE, not the historic NE, and we are interested in some population or metapopulation that is isolated from others, not the entire species NE. What do we have to do if we want to calculate this indicator proportion of populations above NE 500? We need to do two things: define populations and then get their NE estimate. So defining populations is difficult. This will be a big discussion point, I'm sure, but I think it can be done either by focusing on distinct areas, fragmented parts of the range, or for very continuous parts of the range, focusing on ecoregions, ecozones, or other geographic breaks. Then to get NE, as Juan Jose mentioned, there are difficulties using genetic data. So for now, I'm going to talk about using census size data and using the NE over NC ratio. And for trees, as well as many species, we can get census size data from diverse sources, whether that be periodic forest inventories that many countries do. We can estimate NC from area and density. We can get it from published reports or red list assessments, etc., or even by consulting local botanists, managers, and park rangers. And I'll show you examples of those. I want to mention this concept. I'm building on the shoulders of people who came before me. This concept of using non-genetic data to look at the genetic status of species and their populations was proposed by Dr. Sally Aitken almost 20 years ago. So this NE over NC ratio we should mention briefly. With colleagues, including Dr. Rick Frankham, we compiled to the best of our ability NE over NC ratios. There's imperfections to our compilation, but it's pretty good. And as Juan Jose said, most estimates are between 0.1 and 0.3. In fact, 96% of plant NE over NC estimates are above 0.1. So it's a useful, slightly conservative threshold, Terra 3 tree species. There are extreme NE over NC values, but they're basically all in fish. They're not really in our compilation anyways, observed in plants, reptiles, mammals, amphibians, birds, etc. So especially plants, a fairly constrained set of NE over NC ratios. So I'm going to show you some examples of calculating the indicator for individual species. This was the first plant I worked with that inspired my whole thinking on this project. This is from an Endangered Species Act report, which contains information on current and historic populations, compiled by local experts leveraging their expertise. And as you can see in this table, these are the populations and their census size. And very clearly, 4 of those 10 are well above NC 5000, giving us an NE 500 indicator of 0.4. This is a Mexican mountain juniper. We don't have precise census counts, and I want to emphasize we don't have to have precise census counts. We are comparing it to this fairly well accepted threshold. And by consulting local botanists and park rangers, 4 of 11 populations are well above this threshold based on local expertise, giving us an NE 500 indicator of 0.36. This rare oak, native to just a few populations in the US, all of them are below, well below NC 5000. Estimates were made based on recent seed collecting expeditions, and so the indicator for this species is 0. And then the fourth one I want to explain, Santa Cruz cypress occurs in 5 distinct populations in California. 2 of those 5 are well above 5,000, giving us an indicator value of 0.4. So that's just a few examples showing how we do it and getting an indicator value, which can both, again, be used for prioritizing species, which have low indicator values, and for focusing on those populations that are well below the threshold and might be suffering genetic problems. So can this be done at larger scale and for common species? We are still working on this problem, but colleague, Dr. Sally Aitken, has demonstrated feasibility of something similar to this using, again, forest inventory data for more than 40 common tree species in British Columbia. It's a very large geographic area, testing or assessing whether these species are meeting a certain criteria for genetic conservation. That is, at least 3 populations with an NC above 5,000 are protected per seed zone. So using forest inventory data and GIS approaches, this was done for dozens of tree species and found that most, but not all tree species meet these criteria in British Columbia. We are deploying this indicator for hundreds of species worldwide to test its feasibility, along with the other indicators I mentioned. And we're doing this in partnership with the national biodiversity agencies, personnel from these countries to show if it is feasible or not. And I will give you very, very brief initial results. We've been working on this for 1.5 years and just have all our data collected. And this is a very rough summary. Not complete, and it will change a little bit, but not a lot, I think. So far, we have over 800 species. For the populations maintained, we have a fairly optimistic, about 80% of populations are maintained on average for species. But I want to point out, for the species we've examined, most populations are really small. In fact, less than 20% of the populations of all the species we've examined are above the NC 5,000 threshold, and only about 50% above an NC 500, that is an NE of 50. We have quite a few plants in our database. Just briefly looking at the data, I think gymnosperms are in a better condition for both of these indicators. To zoom in on the USA, where we have about 150 species, the situation is actually even worse. Well, it's better for populations maintained, but worse for the NE 500. Only about 9% of, on average, species populations are above NC 5000 or NE 500. So this is the proportion of populations above NE 500. So this is, I think, a really critical observation and a worrying thing for the genetic status of species. So again, I think we need both of these indicators about populations maintained and NE 500. This was a challenging thing. We had to make clear definitions, store data in a standard way, etc. I want to mention a couple of reasons why I think NE 500 is advantageous. It can be applied in all countries using this NE over NC ratio. It's fairly quick to compile if there is previous reports or sources of data on the species. It can empower local people who can be more concerned and even active in monitoring local populations. And it can, again, be explained to policy people. I want to close by mentioning where I think we have a lot of agreement. I think all agree that better indicators have been needed for a long time for genetic diversity than those used in the past. We do need to focus on something to do with populations and not just at the species level, because populations are where evolution happens, and it's also where extinction begins through the loss of populations, and that indicators need to meet these certain criteria. I think we do all agree that genetic diversity within populations is vital for these different processes of adaptation and inbreeding. It's hard, but it's a really important parameter, and we can use NE to get at it. I think, of course, NE 500 is a somewhat arbitrary, round, easy to use threshold. It's imperfect, but I think we would agree that when NE gets less than a few hundred, we are going to run into problems with adaptation, inbreeding, etc. And I hope I've shown you we can approximate NE from NC using fairly reliable ratio. I think we disagree a little bit on whether we need an NE indicator at all. And then I think we disagree a little bit on feasibility. I hope I've shown you a little bit about feasibility of this indicator in my presentation. So to conclude, I think there's definitely a need for assessing genetic status without genetic data. There's an opportunity to mobilize census data or local expert knowledge to get at these indicators. Again, both NE 500 and populations maintained, I think they're complementary and both are essential to get at adaptive capacity of species. And this is needed for prioritization, planning actions, reporting progress, and connecting again to non-scientists. And I hope I showed in our very initial results, indicators can be used to showcase genetic losses and genetic issues that would not otherwise be documented at this scale. Thanks very much.
Thank you very much, Sean. Again, we will take a couple of questions to allow more discussion afterwards. Okay, Robin, please.
Yeah, thanks, Sean. Could you explain how you calculate NC, the census size? So if I go in a forest, I can usually recognize the big trees, but for each big tree, there must be a lot of little seedlings around. And so you get a very different answer, would you, if you counted all the seedlings? Did you have some cutoff? This is supposed to be mature individuals, maybe?
Sure, great point. Yes, this is focused on mature individuals, fairly large, obviously reproductive individuals, so discounting seedlings and non-reproductive saplings. That's a really important point. Again, yes, we're focusing on adult individuals. Thanks.
Okay, thanks.
Thank you. Any other clarification? Santiago?
Yeah, I'm Santiago González Martinez from INRAE, Purdue. I was wondering when talking about any higher than 500, if you have in mind any of the definitions of any of the different ways of, if you think that would apply for better for some of the definition than others, or it's just a generic kind of like estimation?
I would say that the use, when we're using these NE over NC ratios to get at it from the census size, I think this would be a good discussion point, and maybe to hear Robin and Juan Jose speak on this, but I think it's more of a generic terminology and is hard to say that it relates precisely to one of the genetic estimators that that Juan Jose talked about. We're definitely talking about contemporary NE, and I think we could discuss, though, whether what I'm speaking of is more closely related to, for example, the LD NE or the variance NE. I'd love to hear their perspective on that later. This is a really good question. Thank you.
Okay, thank you. I would suggest that we move on to the last presentation from Bruno. So he's going to talk about the limits of effective population size as a practical and operational indicator of genetic diversity trends in forest trees. So Bruno, the floor is yours.
Thanks, Ana Maria. So I will share my screen, and hopefully the right screen. This one. Is it the right screen?
Yes, it's the right one.
Okay, great. So hi, everyone. So my task here in this webinar is to highlight some of the difficulties with using NE as a practical indicator. And first of all, I will start by saying again that we do need genetic diversity indicators. And one of the reasons is that in the CBD, genetic diversity is not really well considered. And there has been renewed calls to say that we actually do need better indicators because genetic diversity is needed for adaptation. We should not just count species, we should not just count habitats, but genetic diversity should be considered. It should be also a target for biodiversity and nature management. And in fact, in the CBD, mostly what you see when talking about genetic diversity is the Nagoya Protocol on fair access and exchange of the benefits arising from genetic diversity, meaning that people think about using diversity and not necessarily protecting it. So first, before going back to Annie, I would like to start again with what is a good indicator. And the question here we have in mind is how well genetic diversity is conserved and sustainably managed. That's the target of our indicators. As Sean mentioned, those indicators should be scientifically meaningful, and they should be able to assess direction of change, so some sort of trends. And the verifiers that we use should be implementable by relevant stakeholders, they being scientists, managers, or policymakers. So indicators actually should be 4 things. They should state the problem. So is diversity being lost in our case? They should indicate why is it lost? What's the pressure on the system? They should also assess what the cost of the loss should be. So those will be benefit indicators. And then they should assess what society needs to do about it. So that's the response indicator. A good set of indicators need to have all 4 of those things together. So I would like to present what is done in Europe in terms of an indicator of genetic diversity. It's part of the Forest Europe, so it's the pan-European management of forest, sustainable management of forest system in Europe, which has created 45 indicators for sustainable management. And one of them, indicator 4.6, is an indicator focusing on conservation and use of forest genetic resources. It has been recently reanalyzed and changed, so it is better, it's more realistic in terms of response of what countries do to protect and sustainably use their genetic resources. So in this aspect, it is not a state, it is not a benefit, it is a response indicator. So a little bit about what this indicator does. It has 4 sub-indicators. One of them I will talk about more in detail is concerned with dynamic conservation of populations of native tree species. Another one in bold type is the potential for production of forest reproductive materials. So we have there an idea of how genetic resources are used for plantation. And then there are 2 other sub-indicators, one linked to the number of non-native species conserved and their populations, so local races of introduced species, and the number of collections of ex situ conservation. So the indicator linked to dynamic conservation of populations of native forestry species has 4 levels. One is simply the number of conserved population, genetic conservation units. The second one is a relative index of species diversity, which considers the number of species which are target species of gene conservation compared to the ratio of the number of species occurring within the country. The third level of this indicator is ecozone diversity, meaning the number of ecozones present in the country versus the number of ecozones represented in the National Conservation Network versus the number present in the country. And the fourth part is an insurance index, meaning the number of ecozones represented by a minimum of 2 conservation units. So really, you can see that this is clearly a response indicator. It uses surrogates. There is no measure of genetic diversity. It uses bioclimate and ecological zoning as a surrogate for gene diversity, which makes sense if you consider that there is local adaptation. And it scales absolute and relative efforts per country at species level across Europe. So with this indicator is really suited to assess what countries do in response for protecting their diversity. Now, if we look at the other sub-indicator, the fourth one that I had highlighted earlier, it has to do with forest reproductive material. In Europe, material that is used for planted is regulated by a council directive and national laws, meaning that every material that is used for planting needs to come from approved material in national registers, meaning that there is a traceability of what is used. However, the way to assess this indicator, so the verifier, is the total number of production units in the country and the total number of species for which there is at least one production unit of forest reproductive material. So it's a very basic way of assessing this response indicator. And arguably, it's not really a way to assess any trends at all in the sustainable use of reproductive material or any risk to its genetic diversity. Actually, this indicator is not really operational for monitoring genetic diversity trends. It's an indicator that's operational for monitoring compliance to law and legal requirements. Actually, few countries have regulations on how they use their forest reproductive material. There's no indication, for example, on how they increase genetic diversity when they harvest seed lots, or there's nothing really related to limit and control of reforestation when the seed supply is low, for example, which would mean a low effective population size in the seed lot. And then there's really nothing in terms of document of seed transfer monitoring. The success also is very low. And for example, something would be very interesting is failure or success of those reproductive material over time when planted. So I would say, given this, the fact that the indicators that we have are essentially related to response, that we do need a state, not to talk about pressure and benefit indicators of genetic diversity. We really need something that tells us if genetic diversity of forest trees is being lost over time and space. For the reasons that were stated by the two, by Sean and by Juanjo, we need to monitor early signs of population decline and extinction, and this would be done easily by genetic indicators, which might not be seen from census sizes or fragmentation, etc. So what we need to know is, do we expect decline, then extinction, from human-made threats such as climate change and fragmentation, etc. And for this, we need to measure genetic erosion. So hence the concept of NE, which was presented earlier. It's a beautiful concept in the population genetic literature, and you could see that it's also a very complex concept. It has many definitions, many ways of being measured. It's probably something difficult to measure for stakeholders, I would say. Also, something that we haven't talked about yet is it's also a concept that comes from the breeding literature, where it's actually the number of effectively breeding males and females. And I think it's interesting to remind a little bit about this. So the idea behind the concept, as previously said, is that if effective population size is low, then there is less potential for adaptive evolution. Then this is an indication of inbreeding threat and eventually extinction. So again, beautiful and something we should consider. Now let's look at the breeding literature and the reason I think why the concern started and the reason why this concept was used to monitor changes and being concerned about conservation and diversity. If you look at farm animal heads like cattle, sheep, horses, pigs, and goats, the graph on the left shows there are tens of millions of farm animal heads in the world, and actually NE, so effective population size, is very low. It's below 400, 500 for most of them. And that's a concern, of course, for breeders and for the meat market and the dairy market, because eventually you might have problems with your herds and you might lose animals and lose potentials for breeding. Incidentally, you can see that the estimates of NE here from different methods in this paper by Hall in 2016 is anywhere between 2 times and 7 times depending on the method used, whether using pedigrees or LD or other methods. So a big concern, you might have huge population sizes, but actually potential for adaptation might be very low. However, so any great, but it has a lot of strong assumptions, which were already mentioned, such as discrete populations, Hardy-Weinberg equilibrium, non-overlapping generations, constant mutation rate, no selection, etc., etc. We find this in nature, already stated by Juan Jose, but I think we need to say that again. In forest trees, gene flow is extensive, also true in fish. The process at work is mostly isolation by distance rather than concrete discrete populations, except for marginal populations and isolated populations, I think, for which we all agree that NE might be a very useful way to assess risk. Trees produce a huge number of gametes. They are gymnosperm mostly, but many trees are dispersed by abiotic processes such as wind. Populations are large, hard to delineate. Generations overlap. There's drift mutation is not at equilibrium. Selection is possibly intense at the very early stage of seedling production. And as shown by my colleagues, estimating any using molecular markers remains a challenge, and particularly true with LD and genomic data in trees which have huge genomes. So in fact, we're stuck with difficulties with the concept, and also because in trees, we might have a problem with estimating NE when we look at sensor sizes. So I will show an example of two trees. So one is the umbrella pine, the Italian stone pine, and on the left with the blue, and the one on the right is the cypress cedar, Cedrus brevifolia. So stone pine is located in the Mediterranean, and it covers something like 600,000 hectares. So it's very widespread, even though in some places it might be fragmented. On the other hand, the cypress cedar grows over less than 2,000 hectares. So you would expect that one is a lot more at risk than the other one. Actually, when you look at NE, it's rather the opposite, with umbrella pine having a very, very low effective population size of less than 10, and actually Cedrus brevifolia having a huge effective population size of largely over 500. So contrary to expectations, the small population size species has higher NE than the largely distributed one. And if you focused on NE, you would protect the umbrella pine and not the Cedrus brevifolia. Although if you think in terms of habitat and threats, obviously Cedrus brevifolia is a lot more at risk than Pinus pinea. Now, why might we have such differences? I want to make a few suggestions and then open the floor for discussion. Perhaps NE, so here it's measured at species level, is somewhat inherited through evolutionary history. So if we look at Pinus pinea and its phylogenetic group, it contains actually a few species which, well, one of them, Aleppo pine, for example, has very low diversity. Okay, a bit higher than Pinus pinea, but still low, Pinus halepensis. And the other ones, well, measured by nuclear microsatellites, you could say that these heterozygosity levels are not very high. And curiously enough, again, the most restricted pine of this group, Pinus canariensis, is actually the one with the highest diversity. So perhaps something to do with phylogenetics over a long period of times. Perhaps something that has to do also with ecological requirements and the fact that in Europe, the Pleistocene climate changes have affected very strongly populations and species that grow at low elevation that are very thermophilic, like Aleppo pine and the umbrella pine. Something to think about. Another suggestion coming from this paper is that when you compare the umbrella pine with maritime pine, Pinus pinaster, the difference in NE is something like over 200 in the ratio. There's no indication of threat in any of those species. And if you look at the genomic composition of Pinea compared to Pinus pinaster, well, there are actually fewer highly deleterious alleles in Pinus pinea, and the deleterious alleles have been purged from regulating and stress response genes. So actually, we may have a system there where some mechanisms prevent the loss of adaptation, although effective population size is very low. For example, in islands of diversity in the genome, and also through increased plasticity. And I think their plasticity is also a component that we need to consider when creating indicators of state of genetic diversity. Three other things that I would like to put in the discussion here. Well, actually, I haven't been able to find, but maybe I haven't looked enough of actual link between low NE and some sort of extinction vortex in the wild. So except perhaps for the Florida panther, where when you remember the story, the Florida panther is almost going extinct because of inbreeding and small population size. There are 26 remaining individuals, and the translocation of 8 females from Texas was enough to restore the population to a high level of diversity. So it's very small numbers that make it possible to retain diversity and potential for adaptation. I would also think that perhaps census size and not other levels, other measures are the primary concern and factor of population extinction, because this is where human actions actually are the strongest. And another thing I was wondering about is hybridization in conservation is often considered as a threat because it means genetic pollution. Obviously, in the case of the Florida panther, it was considered not as a threat. But with genetic pollution, of course, NE would increase and you wouldn't be able to measure that. So in the end, NE is a complex concept. Again, I think a beautiful one, very useful in population genetics. I think it has one name and too many meanings for now. It should be cleaned up. It's measured in very many ways. It's often unresolved using genomic data, and sometimes the relationship between census size and effective populations are not so straightforward. And perhaps it might be hard to involve stakeholders. And I think here we need to be careful because I find it, I don't know about others, but I find it that involving stakeholders over notions and concepts of genetics and adaptation in a genetic sense is very difficult. So we need to find ways to involve stakeholders and embark genetic diversity in conservation and indicators. So NE could have merit as a state indicator. Obviously, we are needing some, at least in Europe, if estimation methods were clarified, measurements standardized, perhaps within phylogenetic groups, within species, if trends were reported and not just single measures. Perhaps other candidates of such indicators could be other widespread measures such as heterozygosity, allelic richness, and of course, the use of ecological and phenotypic proxies from forest inventories and remote sensing would be a very sensible as well, such as age class, regeneration, perhaps rearing variants, seed set, etc. And I will end with a nice picture of a tree ring sample showing tree ring with differences and the fact that those are systematically taken by national forest inventories. And I will stop with this. Thank you very much for listening.
Thank you very much, Bruno. As we did before, we will allow a couple of questions now for clarifications, and then we will move on to our open discussion. So I see David Bossier.
Thanks very much for your talk. Very interesting. So are you suggesting that rather than being worried about absolute levels of NE, we should be concerned more about the change in NE in any particular population or species, which of course raises issues of the difficulty of actually measuring changes in NE?
Yes, exactly. I think it's to be able to measure any change and offer ways to respond to those change and modify practice. I have assumed in my talk that it's human interventions that actually are mostly the threats for populations. It's changes that we need to assess. So, for example, moving from any measured at a adult population and any measured in seedlings might be something that is interesting. But as mentioned earlier, it's a challenging thing to do, and the number of samples needed to do this and so on is quite complicated. But an indicator that can be remeasured over different periods of times for me makes more sense. That and even if it is a state indicator, it needs to be able to be measured over time.
Okay, thank you. Now I will ask the three presenters to turn on their cameras so that we can sort of have like the panel in our screens. And I would like to open the floor, so you can raise your hand or type your questions in the chat. Santi?
Yeah, I was wondering, like, if, because in some of the discussion I see a semantic problem. So for me, for instance, like, and maybe we will solve many issues if instead of calling getting an indicator that is effective population size, we'll get an indicator that is, I don't know, 10% of census size, or the number of reproducing trees, or number of reproducing individuals, or a particular something that is really, really connected. I mean, we don't talk about process or something, but we call things by their name. So it's totally connected, the way you measure and estimate it with the way we call it. So that's a general reflection, because I think I find that the main issue is actually into the how to estimate effective population size, if that is possible. But at the end, it's reduced to census sizes. We could call it like that, right? So that's a thought.
Yeah, cool. So maybe I can answer. I would agree. I think the breeders have that in mind when they count the number of reproducing individuals and say, okay, our effective population size is, despite the fact that we have millions of heads of cattle, then we have an effective population size of 100. So that's the number of individuals that reproduce. And I think for a census, for creating an indicator, that would make sense. Now, it's not completely straightforward what you count as a reproducing individual. I've had the challenge in some of the gene conservation units we have in France, where when you go on the field and you say, okay, what is a reproducing individual? Is that this small tree with 3-4 cones, or do you only have a huge, big individual with hundreds of cones? I'm talking conifers because that's what I know best. But so it's not completely straightforward, but I think it might be easier to grasp for people on the ground, for those who make the who manage, who monitor, And it's certainly a lot less confusing than effective population size, which, as we have seen from the presentations, can mean a whole range of different things and still have the same name.
Thank you. I see 3 hands. Aline,
Ana Maria,
you can.
Do you want all of us to respond to this one?
Yes, of course. I just wanted to mention that there are people waiting on the line, just for them to know that I have captured their names. Please go ahead, Son.
Thank you so much. Yeah, this as Santi said, it's somewhat of a semantic issue, what we call this concept, and focusing on NE 500 or NC 5000 or some other similar number. My colleagues and I went with effective size when we were speaking to the CBD stakeholders for I would say two reasons. One is because we wanted to make it clear that we are focused on genetic diversity. So if you start to talk only about census size, people will then say, well, we're not even talking about genetic diversity anymore. So we wanted to make it clear that this is really about effective size and genetic diversity. Another reason was we do want people to be able to make use of knowledge of the NE over NC ratio differences among taxa and to use that range. So although NC 5000 is like a default, it's a bit of a conservative default. So it would be possible and even encouraged to use maybe both the whole range. So report the indicator under the condition of NE over NC 0.1, and then report the indicator under the condition of NE over NC of 0.3. And that gives you a full actually documentation of uncertainty in the indicator, which is actually really valuable, I think, for stakeholders. So in some of the examples I showed, if you use a different ratio, you get a slightly different indicator value, especially for the Santa Cruz pine that I talked about. So that's another reason to use the terminology effective size. And then lastly, in terms of the difficulty of the concept, for stakeholders, I want to emphasize they don't have to understand the full complexity of the concept. If you try to explain the concept of a population to a non-ecologist, there's a lot of complication in the concept of a population or of other things like community structure or extinction risk. To fully understand the PVA under an extinction risk analysis is actually very complicated, but you can explain it very simply to a stakeholder without going into all of the details. So I want to emphasize we don't have to consider explaining all of the details to stakeholders. So that's all.
So I would just add, if I may, to that I think both could be useful. For sure, I think that the census size, as Santi said, has to be reported because in many cases it's probably the most robust estimate that we will get for both demographic and evolutionary processes. But on the other hand, it's at the end, it's a parameter that determines mostly demography. So of course, if we are dealing with genetic resource conservation, we need something more in addition to the census size. But of course, there is complications there. First, in the definition of census size itself, as we mentioned before. So it will be clear for a discrete population where the limits are evident. But more generally, what happens if you have a genetic conservation unit that has a given size, but it is within a much larger population? So there, what is the census size you should report? I mean, of course, the local one, it's meaningful because it's the one under management and you should report it. But then I think we need a bit more information about the surroundings. You need to report whether this is part, I don't know how this could be done, but you could talk about a number of trees that are present within a given radius, for instance. If you have tens of thousands of trees around your population, probably your effective population size is going to be even larger than your census size. So I think we need to report census size of the administrative, let's say, limits of the population, but also even something else about the truly biological population there, and also ideally, of course, about whether there might be some gene immigration from other more distant populations. And this rule of thumb of the 0.1 census size, I think can remain useful in addition to the census size, but knowing that this is a general estimate that, of course, may not always work properly, but it's something that for managers, I think it can be useful, because otherwise we certainly in general need more individuals than the census size to maintain evolutionary potential in the long term. So.
Thank you very much. If there are no more comments from the panel, I would move on to the next question now. I have Aline Finger.
Hi there. Yeah, thank you very much, everyone, for really nice talks. That was super interesting. And yeah, I'm Aline from the Botanic Garden in Edinburgh, and I would just love to hear your thoughts on clonal species. So I don't work on forestry, so that might be a random question, but how would you deal with populations that have a lot of clonal growth or rhizomal growth? Because obviously, I mean, you may have a low number of genetic individuals, but they have an increased likelihood of having sexual reproduction if they flower a lot. They have large individuals. So I was just wondering whether you have any thoughts on that, and in terms of extinction risk as well. It's quite different.
Yes, John, please.
Thanks, Aline. I have also worked on some clonal individuals, and simply every genetic replicate that's a part of a clone would all be considered one individual when you're talking about the effective size or even the census size, truly. So for a clonal individual, I've worked on clones that can be tens to hundreds of meters, not much bigger than that, but in such case, you either need someone with good biological expertise who can tell you how big a clone can be, or you need some genetic data to determine how big clones can be. And then in that case, you would probably be using some estimate of the area of the site divided by the average, area of a clone, and that would give you a rough approximation of the number of genetically unique individuals.
Anyone else from the panel that would like to comment?
Yeah, also maybe in that case, I think it's one of those things with the census size. I think it will be mostly very interesting, because especially for these species that have huge clones or extending over big areas. I mean, these are probably species that are adapted to develop their demography, even with very low genetic diversity. So if you get estimates of any for those locally, you probably are going to get tiny numbers. But for sure, what we see is telling us that these species can do okay with those locally, with those tiny numbers. It's more the demography and how they manage to propagate that matters in that case. So in those cases, I would rather look at things like just survival and recruitment, and because the evolutionary process may be very different from other sexual species.
And perhaps then if census population sizes are very small, then the ex situ strategy would work. It's true that I have in mind the example of this very rare cypress that grows in the Sahara Desert in Algeria. It's a Tacilis cypress. It has evolved a very strange sort of clonal propagation where pollen is not reduced and germinate in the ovules, such as would be in a surrogate mother kind of system. And the individuals are highly heterozygote. None of them are homozygotes at all of the loci that you can measure. And so it's really a clonal system where high diversity is maintained, although the census size of the population is really low. So perhaps clonal populations have indeed strategies to maintain diversity.
Great, thank you.
Thank you. If there are no more comments from the panel, I will move on to the next two hands raised, and then I will also check the chat because we have some questions there. So I'll give the floor to Robin.
Okay, thanks. So all of the speakers pointed out some of the complications with multiple definitions of any, at least half dozen or so in the literature. I mean, they're confusing even for scientists, let alone stakeholders trying to explain to them. But I think with only a risk of a little bit of simplification for practical application here, we can reduce this to basic two kinds of effective size. One is the local effective size, contemporary local effective size. That's where that number then reflects demographic genetic processes that are going on currently within the local population. And they're determined by the distribution of offspring number among the local parents. So that's a demographic process. It can be detected genetically by things like the LD method or even the temporal method. The standard temporal method is also mostly sensitive to what's going on locally. So on the one hand, then you have local effective size, and you can measure that or estimate that both demographically from the pedigree and the distribution of offspring number among the parents, and you can monitor it genetically with say like the single sample or temporal estimators. Both of those should be giving you roughly the same answer regarding local effective size. On the other hand, in contrast, you have a global or maybe even species-wide NE, or at least metapopulation NE, that is very sensitive to even low levels of migration. So this, excuse me, metapopulation NE generally will be larger and often much larger because it integrates local effective sizes across the different populations that are interacting by migration. And furthermore, this metapopulation NE also operates on a longer time scale because the effects of migrating alleles, even very low levels of migration, like one migrant per generation or less, are sufficient to keep most of the alleles circulating within a metapopulation over long periods of time. So this metapopulation or global sort of NE concept also has a deeper time element because it depends on processes that occur over many, many, many generations. So those are two big contrasts between the local and the metapopulation or global. And I think that they potentially could be explained even to stakeholders as something that they could understand. And it's not the case that one of them is scientifically better or right or correct. It's that both are potentially valuable. They give you different types of information. For example, in the example that Bruno gave with the the single population of a cedar in Cyprus, I think, and then a widespread pine species with estimated low NE of something like 20. That must be a local, either a demographic or a genetic estimate of local NE. So to me, that would suggest you have lots of local populations, each of which might be fairly small, but collectively, the amount of genetic diversity in that species might be really high, and it might be pretty high within each of the local populations. I'm not sure, because I'm not sure what sort of data were used to generate those low estimates, but it could be that the low estimates apply to, say, an estimate that's just estimating local processes, but in that case, could be misleading if you think it's telling you how much genetic diversity those local populations have. So to me, that is the biggest distinction to make in terms of practical application of the NE concept. It's whether you're talking about local NE and then what's going on with the most recent few generations, sort of contemporary NE, that covers about the time scale of your samples versus metapopulation or global NE. That applies to on a much bigger scale. It's affected by very low levels of migration. And it reflects processes that have occurred across many, many, many generations. So there's a big inertia in that system. To me, those are the two most important ideas to keep in mind in trying to apply this concept to real world problems. Thanks.
Thank you. Who from the panel wants to go first?
Well, maybe me, Bruno, for clarification. Actually, the NE for the Italian stone pine is a global NE, so it's very peculiar. And the interpretation for this species is that during the glacial cycle of the Pleistocene, it was reduced to possibly just one population somewhere on the Mediterranean coast and then recolonized from there. So hence the super low NE across the range. The estimates of NE were made using genomic data, multiple microsatellites and SNPs from genomic regions in the species. So it's a peculiar thing. It's rare in most tree species where the NE in conifers is often a lot higher, and that's why I gave the example of this Cyprus cider, where census size is low, but still it was able to retain quite a large effective population size. So that's the precision I wanted to make. But otherwise, I agree with the two types of measures.
Yes, so I think Robin's points are excellent. I mean, maybe we are thinking too narrowly in terms of a single definition of effective size, and as we are seeing and as people are showing, and as Robin mentioned, actually different evolution. As soon as you don't have a single isolated populations, we have different genetic and evolutionary processes operating at different spatial scales. So it's clearly meaningful to have at least two estimates of the census size as soon as we don't have an isolated, truly isolated population. So I think it's a very good idea to propose this, to having two estimates, one local and one meta population. So of course, then the issue is how large the meta population to be considered should be. and this will depend on the dispersal range of the species. So, but I think clearly we cannot neglect what is around the target population in terms of other conspecific populations that are potentially within reach of gene flow. And then if we report these two estimates, managers can consider them. We can inform them that things like variance of allele frequencies or linkage disequilibrium normally operate locally, but other things like inbreeding or the maintenance of additive genetic variants, they typically are influenced by the metapopulation process as a whole. So yeah, I think that's a very good point that opens additional also difficulties on how to define the metapopulation meaningfully, especially in forest trees where you have potential for very long range dispersal.
I would only add, yes, I think it's useful to think at the local and then at the metapopulation, possibly at the global level for effective size. I would zoom out to a broader level and think in terms of practically applying this for conservationists. The local level, some people have said it's most important to focus on this NE of 50, so for inbreeding, and maybe more at the metapopulation level for NE 500. So some people have discussed that in the past. I think either is fine. I think my main takeaway and emphasis is just to not forget that the something with effective size is important. So just to not entirely neglect this aspect of populations. Maybe we're maintaining populations in a region, but if those populations are very small, then they are suffering some genetic consequences. So we can just be pragmatic and keep it simple as possible, but still make sure we are ensuring that, especially for range edge or separated populations, they're of sufficient size. There's a lot of talk of widespread species over millions of trees. Great, they are very, very high above the NE 500 threshold. We don't need to estimate the exact size. We know they are very high above it, so they are checked. They are green. Policymakers like things green, yellow, and red for warning. So we maybe want to keep it somewhat simple in those terms.
Thank you. Then the next one is Ivan Scotty.
Hi everyone, great talks. Thank you very much. Well, I'm Ivan. I'm from the same lab as Bruno, so I'm not going to give details about where I'm from. A couple of comments. One is about the sensor size as an estimator, which is absolutely fine to me. It's okay. It's great. I was wondering that, of course, that using the NENC ratio somehow behind that is the idea is that you have for a given species or population a constant ratio, because that's the way the population works. I mean, that's the number of individuals who reproduce and so on and so forth. What happens, but what happens, and I mean, the interesting moment is when that ratio changes, right? I mean, if there is something that changes the ratio because only a subset of individuals keep reproducing, and that's what you are really concerned about in terms of conservation. So maybe a comment on that. But of course, this is particular just to Sean. But and the other comment is okay. But I think the starting motivation of all this discussion about forestries is that very often what we do is as we have conservation units, which are maybe a plot of few tens of hectares, which is part of a way larger area. And so we focus on computing NE or whatever it is in the conservation unit. And that may, as we have been discussing today, only reflect the local neighborhood size, actually. And we can be happy with that. But of course, we don't buy it somehow. I mean, if we know that the conservation unit is 10 hectares, it is embedded in a 2 million hectare continuous population, nobody will believe that the effective population size is the neighbor size within the conservation unit. So we're still left with the same problem. What do we do? Do we multiply neighbor size times the ratio of surface of the conservation units and the global forest, or what do we do? Thank you.
Thank you, Ivanson.
I have two very short answers. For the NE over NC, I acknowledge and I emphasize that it does vary among populations, and you're right. The meta-analysis I presented somewhat collapses variation across populations. That was just averages at the species level. So I think we need further compilations and better understanding of how it varies among populations. And I think that kind of publication and analysis is totally missing in the literature and is needed. Simulations can also be used to show how population level NE over NC might differ. As for conservation units, if I were to propose an indicator or a requirement for NE relating to a conservation unit, I would say that the conservation unit would have to have a minimum NE of 500 as a part of the forest that it is a part of. So some of those conservation units might be small, isolated fragments, and in that case, it's just that unit. But the whatever component that unit is participating in, gene flow, would be what NE has to be greater than 500, in my opinion.
Thank you. Anyone else from the panel that wants to reply? Juanjo?
So yeah, I think that those are two excellent points, Ivan. And as well, as Sean has said, I think what should we do? Actually, one common sense thing is that, as someone said before, that if we have a huge population, actually the conservation concern is probably minimal from the genetics point of view, and it's moreover it's a case where the estimation of any with genetic methods is much more difficult because the signal to be captured is minimal and it's dwarfed by sampling noise. And not only that, but the variance of the estimates is huge, so that you really cannot look at a trend in time, because any potential trend will certainly be much smaller than the variance of the estimation variance itself. So yeah, for huge populations, I think the answer is, well, if population is huge, you'll be happy with it. Just keep looking if the demography is changing, or if you have recruitment especially. And if you have, then why bother about the rest? Invest your efforts in smaller populations and preserve the habitat, and that's what you should do. Okay, so and in the specific case of a genetic conservation unit, it's a bit like that. I mean, as Sean said, well, you could say for sure you should have at least, for instance, 5,000 trees, assuming that's the census size, that you have an effective size of 500. But of course, that will be a minimal estimate. I think we should also report the census size of not of the genetic conservation unit itself, but of the surrounding population. If it's a continuous population, give the number also of trees that are in the surroundings that make a continuous stand, because that will be more meaningful. I mean, and then is the issue, of course, of the metapopulation that we mentioned before, that's more difficult to define. And also the other point you made is very good. Okay, so we are looking at trends, we are looking at changing conditions, so probably any is changing. Maybe the census size is not changing, so the ratio then should be changing. And if we are assuming a constant census, but okay, so we should make clear that this 15,500 rule it's a rough estimate, and that we are giving a minimum probably estimate, and this does not serve to actually to finally monitor the trends because maybe it's 0.1, maybe it's 0.5, or it's 0.3, and maybe the variation per generation it's 0.001. So I mean, we really cannot look at those changes, I think, unless they are really dramatic in a very small population, in which case it's much easier to estimate everything.
Bruno?
Yes, in fact, with widespread tree species, one of the issues is what happens at the margins. Particularly with climate change, we have margins that are under high threat, and for those, estimating a local NE versus global NE and having some sort of trend, I think would be something hugely important. And as a matter of numbers, for example, the rules for the gene conservation units in France for those with widespread populations is to have plots that are at least 500 individuals, reproducing individuals. So we're far from 5,000. But as you say, since they're surrounded by a lot of other trees, then maybe it doesn't matter so much. And then some GCUs were located at the margins of distribution areas, where there you take all the trees that you can find, and that's your GCU. So in this case, you will have possibly a local effective population size, which is much lower than the global estimate. So perhaps relating those two estimates, as previously stated, might be something, and doing that over time is obviously something that we should think about.
Thank you. Then I think that we are like running out of time. We are close to the end of this webinar. I will allow Luis, and after that we will conclude with some final remarks from the panel. Luis, please go ahead.
Hello, my name is Luis. I'm from the EUFORGEN focal point of Switzerland, and I've a practical question to Sean. Actually, you were discussing about the difference between the census size and the effective population size, and that some trees could count to the effective population size, others not, and this is not so easy to which one you count to the effective population size. And you said that you did estimations of both, but these were with inventories and with botanists that were basically getting this data. How did they make the difference when a tree belongs to the effective population size and when not?
Thanks for the question. For forest inventory data, this is often done using a minimum diameter threshold, which is imperfect, and it doesn't mean the species is an adult or even that it's reproductive or anything, but it's the best we can do. So that just means it's of a certain size, usually no longer a sapling and thus somewhat reproductive, but not always the case. So there's a lot of imperfection, but that's usually what's used. And then for counting adults, kind of just by local experts or local knowledge, that's often just the trees you can see that are fairly obvious, and usually that will be a much bigger DBH. You might miss some of the smaller ones. So there are definitely some issues with that, and if we are to go forward and use this in the future, we would need some better definitions of what should be counted as an adult. And although I would emphasize these are approximations, and again, we are interested in really just general trends of is it very much above or below these kind of numbers. So exact counts are not as needed, but I agree we would need some more standardized definitions of what an adult tree would be considered.
Thank you. Any other comments on that from the panel? No. If not, I would like to ask in the order of presentation. I will start with Juan Jose, some concluding remarks for the webinar.
Okay, so maybe main conclusions I would think is, I would like to say it's we need to talk a bit more, I think. That's because it's been very fruitful, I think, this discussion. And then, well, I think there are two main things we have to consider in practice. I mean, when we are dealing with small, isolated populations, everything works quite well. We don't have to care so much how the precise definition and estimation method that we use. Most of them will converge, provided we have enough sample sizes, which will be achievable in general. And the problem is, and well, the good news is that those populations are the ones with greater interest for conservation. And then the separate thing is that when we have large continuous populations, as is the case frequently in trees, and in that case, we should be aware that for management, we should be careful of what we propose, because in terms of accurate estimation, we should not promise more than what we can deliver. So using genetic markers has great potential, but also have many complications. And managers should be aware of this. So it will be important to think in terms of both local and regional distribution of individuals, and beware that some of the process of interest in conservation may not necessarily be operating locally, but rather regionally, and that you should not neglect the distribution of individuals outside and the potential gene immigration from the outside into your target population.
Thank you very much. Then, Sean.
Thanks. I think reflecting on Juan Jose's talk, there are still cases where genetic markers will be very useful for estimating effective size. In certain cases, especially isolated populations, as he mentioned, there is a need to emphasize, the confidence intervals of those estimates and other issues, but it can still be useful, especially as Ivan Scotti mentioned, there are populations that will diverge from this N/NC ratio due to bottlenecks or some other issue, recent colonization, etc. So genetic estimates are still useful in some cases. In my opinion, in the majority of cases, due to cost and due to just practically getting information quickly and rapidly to managers and policymakers, we probably want to mostly emphasize the census size and use a general N/NC ratio. I suggest calculating both at the 0.1 and 0.3 ratio to give some measure of uncertainty, which I think is valuable. So that's a positive and a more conservative viewpoint on the population status. And then again, if we were to use this practically for gene conservation units, we would consider the NE500 or the NC5000 or some other threshold at a local level if it's an isolated population. But then, of course, yes, if the gene conservation unit is embedded within a large forest, well, it's already clear that that forest is probably well above the threshold. And again, I want to emphasize we don't need exact counts. Someone mentioned earlier, change in NE. I think monitoring change in NE is important, but also really this threshold of NE500 is important, even at a single time point estimate, because it signals genetic erosion that has been happening. So I want to emphasize that as well.
Thank you. And Bruno?
Yeah, well, first of all, thank you for the webinar and for inviting us and having a very nice discussion over these difficult concepts. I wish geneticists would call things more simply and not mix things together and when actually they mean different things. So that would be one of my suggestions is to, well, maybe our local NE and global NE might be something nice to add to NE to make things clearer. What I would like to state again is that, at least in Europe, we do have very nice response indicators of how well genetic diversity is conserved and sustainably managed. We lack state indicators of is diversity being lost or not, and that's an important thing to have. So that's one point. And the second point is that we need to be able to monitor trends. So states at different time points is Sean said, at least have a state is important, but also trends in those states is quite urgent. And we should not forget, even though some tree species have huge populations, that they may soon have very small populations in many parts of their range because of climate change. And so in species where we think that risks of loss of adaptation and loss of diversity is not currently high, we still need to monitor those and to make sure that we understand the processes going on. Because with Europe is currently on a +4 degree increase in temperature over the 21st century, as said by our politicians. So this means that a lot of tree populations, even for widespread trees, will have to, will suffer serious threats. So indicators that understand, monitor, and show genetic diversity changes are definitely needed. Thank you.
Thank you very much to all. I think that, yeah, we still need like a lot of discussion. But after this webinar, I think that we had the time to understand each other better, to address this issue. And of course, yeah, we still have a long time until we finalize and we identify like the best indicators that will fit every single case. So with this, I would like to thank all of you, to thank the panelists for joining us in these very nice presentations. And also I would like to thank all the participants for being here with us today. It was really nice having you all. And I hope that we will see you soon at another occasion, at our next webinar. Thank you very much, everyone. Bye.
Thank you, everyone.